A very good afternoon, dear students. I am Professor Dr. Vikas Mishra from Department of Microbiology at GSJMU Medical College, Kanpur. And today in this lecture we will be discussing of isolation of bacteria in pure culture form. So first of all, you need to know what is a pure culture. A pure culture is a culture which contains single species of microorganisms. Therefore, aseptic technique must be followed for culturing of that culture media. There are a number of procedures to obtain pure culture, and it is very important that it is restricted from contamination of other bacteria. We should isolate the same bacteria which we have cultured in that recovery medium. So first the question is, why pure culture? Pure culture is used to identify and study the characteristics of that particular microbe which we have isolated in the culture. Also to detect the causative organism of a particular disease. So medical samples from clinics, samples associated with food products and the environmental samples contain mixed culture of microbe. So it is really very difficult to identify each microbe. Therefore, you require to isolate the colonies from there and prepare a pure culture form so that you can further test the organism on the basis of biological, biochemical test as well as serological test. For example, if we take 1 gram of feces, it has over 10 to the power 10 millions of bacteria. So you can very well appreciate that what is the burden of bacteria in that simple 1 gram of feces, and that too, that 1 gram of feces contain 20 different bacterial species. Therefore, to identify the bacteria and antibiotic susceptibility of that bacteria, it should be isolated in a pure culture form. Therefore, pure culture is highly essential or important. Talking about history, the technique was developed by German bacteriologist known as Robert Koch and his associate. So, the earliest pure culture form was developed by Robert Koch. What did he do? That a potato was sliced with a sterilized knife which was heated, and then it was boiled and inoculated and streaked the bacteria over that potato surface, and then it was incubated beneath the bell jar. Unfortunately, that first pure culture form, bacteria did not grow well, but that was the start, that was the history. Sir Frederick Loeffler, who was an associate of Koch's, developed a meat extract peptone medium. He spread a mixture of Loeffler's medium and gelatin over glass plated and allowed it to harden, inoculated the surface in the same way as potato was inoculated. The gelatin was melted and some were eaten by the bacteria. It was utilized for nutrition by the bacteria. Minora Tarazimon in 1882 discovered agar, which is used now as a solidifying agent, and this was developed from extra seaweed soup. It was used by the East Indian Dutch to make certain jellies and jams. The Jersey, who was the wife of Walter Hesse, Koch's assistant, learned of agar from a Dutch acquaintance, and she suggested to use agar as a solidifying agent in the culture media. Certain aseptic techniques are used to identify or to sterilize the culture media. Use common sense to avoid contamination. First of all, you should have common sense that by doing that procedure, you should not contaminate the media. Certain non-sterile objects must not touch the culture media and other equipments which you will use for preparing the pure culture form. Then, do not remove the lid of Petri dish completely. Just lift it and hold it above the dish. There is a procedure which you will be demonstrated in practicals, that how you don't have to completely take out the lid. You have to just hold it upright in a 45 to 60 degrees angle and inoculate it in order to avoid contamination. Also, you have to remember that do not put test tube caps or petri dish laid down. Sterilize your inoculating loop or needle by heating it until it glows, and then allow it to cool, and then you take the inoculum from the sample. So if talking about the method for isolation, there are two methods for isolation. One is plating method and the other is most portable number method. Talking first about the plating method. It is the most common method. Usage of a Petri plate is required, and there are three types of Petri plate method. Number one, streak plate method. Number two, spread plate method. And number three, pour plate method. These are the culture methods which are utilized for inoculating the bacteria or the isolate on the culture media. So first, the streak plate method. This was developed by Loefflers and Gefke in Robert Koch's laboratory. Continuous dilution of the microbes by streaking results in separation of individual cells. The mixed microbial culture is transferred to the edge of an agar plate, and then a series of parallel non-overlapping streaks are performed on that culture media in order to isolate bacteria. So there are different types of streak patterns. You can have a single streak, you can have a continuous streak, you also have a T streak, then radiant streak and quadrant streak. So these are the examples of various streak methods which are employed. You can very well see in this picture. And this is how after incubation in the incubator you get the colonies isolated. This is the T method and this is the separate plate method which is shown in this picture. This is a continuous streak which is shown. Now how it is performed? The streak plate method, I told you, ideally the first you have to put the culture on the edge of the Petri plate, then gradually you have to turn 90% clockwise. So the first is the well you prepare and then you turn it to 90 degrees anti-clockwise, move aside and put 3 individual streaks there. Then again you rotate the Petri dish and again you put the 3 streaks. Then again rotate and put 3 streaks. And ultimately the last, which is the tertiary streak, is done. And this is how when you incubate you see the isolated colony. If you see initially the colonies are together, so you will not appreciate the colony morphology. But slowly and slowly as you turn in the primary streak, secondary streak, tertiary streak, you are going to isolate the colony respectively. They are more further appreciated from each other. Then we have a streak plate isolation method. In this, as I told you, first we take the inoculation loop, we sterilize it red hot by the help of Bunsen flame firmer, and then we take the loop, allow it to cool, insert it in the specimen, take the inoculant, and we put on the culture media first set of streaks. And please remember, at this time you need not open the lid and put the lid away from the culture media. You have to just hold the lid up to 45 to 60 degrees so that it is not contaminated by the environment. Then you gently rotate it and put the second set of streaks. Again rotate it and put the third set of streaks, and lastly you put the fourth set of streaks. After this inoculum, you again sterilize the inoculation loop and put it back in the inoculation loop holder. You close the lid, and with the lid without opening it, you put it in incubator for overnight incubation at 37 degrees temperature, which is the room temperature, which is the temperature of the body. Now, after overnight incubation, you are going to see development of colonies. As you can appreciate, the primary streak, which is the first set of streaks, there the colonies will be too much together. So you are not going to appreciate the colony morphology. Slowly and slowly, besides second streak, third streak and fourth streak, you are going to see colonies are going away from each other. And at the fourth set of streak, you can appreciate each and every colony and you can study the colony morphology. So this was for the streak plate method. Then we have a spread plate method. This spread plate method is suitable for obtaining pure culture isolation. It is done for the microbial suspension, which contains about 30 to 300 colonies over the agar plate. It is always done by a sterile bent rod or a spreader or a L-shaped rod. But before you start the procedure, you should always sterilize it before using, so that it is not contaminated and you get the pure culture isolate. So how it is done? What is the procedure? The first step is to transfer about 0.1 ml of dilution of the serially diluted sample on the center of the pre-poured agar plate. Then it is slowly and gently spread over by the L-shaped rod, and it should be so gentle that you do not disturb the solidifying agar there. You do not press, you do not apply pressure so that the media is destroyed. After spreading it, you incubate it at the desired temperature for overnight, and then you calculate the number of colonies which are present there. And this type of method is very useful for the heat sensitive microorganisms. So as I was telling you, this is the spread plate method. The first step is sample is pipetted on the surface of agar plate. It should be in the quantity of 0.1 ml or even less. Then sample is spread evenly over the surface of agar using a L-shaped rod or a glass spreader. After that, you close the lid and incubate it. Then on incubation, you will see the surface colonies. These surface colonies can be visualized and number of colonies can be counted easily by this method. And as I told you, for heat sensitive microbes, spread plate method is of choice. Then we have a pore plate method. In pore plate method, original sample was diluted several times. The diluted sample of 0.1 ml or 1 ml was mixed with liquid agar at 45 degrees Celsius, and then it was poured on the Petri plate. After incubation, the colonies appear on both within the agar and on the surface of agar. Why? Because you have already mixed it in the liquefied media. So it is homogeneously spread. The sample is homogeneously spread in the media. And once the sample is mixed in that media, you pour it. And after pouring, you place it for solidification and then incubation. So after incubation, the colonies will appear both within the agar and on the surface of agar. Now, for following microorganisms it is suitable, that is for aerobic, facultative aerobic and non-stringent microbes. This pore plate method is of choice for culturing the bacteria. And here a slightly more inoculum, that is more than 0.1 ml, can be used. And yes, this method is also not suitable for heat sensitive microbes, and it is not suitable for diagnostic state. This slide shows you that after mixing the inoculum, you are pouring it into the Petri plate, allowing it to solidify at room temperature when the lid is closed. After some time, without displacing the media, you straight away put it in the incubator for overnight incubation, and after overnight incubation you are going to see the colonies which are present within the agar as well as on the surface of agar. So this slide also depicts the same method, that serial dilution was taken, 1 ml was transferred to the diluted bacterial suspension was taken in the primary test tube, then 1 ml was transferred into the another test tube which is having 9 ml of molten agar medium, and then this melted agar medium in this diluted bacterial suspension which you took has homogenized, it has mixed. Then you pour this test tube on a Petri plate and incubate it for overnight. After incubation, you can see that solid agar medium, you have the colonies both on the surface of agar as well as within the agar. Now we'll be discussing about most portable number method. It is one of the type of pure culture isolation. It is used for the estimation of number of microorganisms in food, water and other samples. Even milk can be used. Mainly done to determine the coliforms and especially Escherichia coli if it is present in the sample or not. And this test is designated by letters MPN, which means most probable number method, MPN method. The principle lies that coliform utilizes the lactose which is present in the medium and it produces acid and gas on fermentation. There are 3 phases in this. Number 1 is presumptive, number 2 is confirmatory and number 3 is completed. This slide depicts how you perform the test. First, you take the original sample. If you think about water culture, suppose you are taking the sample of a pond or a lake. So, original sample is taken in a flask, then it is diluted in serial dilutions. How it is done? First, you pour 10 ml in series of 5 test tubes, then you pour this 1 ml of this original sample in series of 5 test tubes, then the quantity is further decreased, it is poured in 0.1 ml of 5 series of test tube and inoculate 10 ml in each tube, inoculate 1 ml in each tube, inoculate 0.1 ml in each tube, which is containing equal amount of culture media. So what you have done is that you have serially diluted the pond water sample in a desired culture media ranging from 10 ml to 1 ml to 0.1 ml in a series of cultures tubes, and you also put inside that liquid culture tube Durham's tube. Durham's tube is put always inverted, so that what happens if the bacteria is utilizing that sugar solution lactose, it will produce acid and gas. That gas will appear in that inverted Durham's tube at the top in the form of air bubble. So after incubation for 37 to 24 hours, what will happen? These test tubes will have, which have sugar in that, the bacteria which is present in the pond water will start utilizing that lactose. Therefore, more acid will be produced. If more acid will be produced, the pH of the medium will go down. When the pH of the medium will go down, the color of that media will change from red, pink colored to pale or straw colored or yellow colored. It is only due to fermentation of sugar lactose and production of gases. And of course, you will see that if the bacteria is also producing gas, it will be in the form of air bubble which is present inside the inverted Durham's tube in that liquid culture media. So as I was telling, the first phase was presumptive phase, in which 15 test tubes, each 5 are inoculated with 5 ml, 1 ml and 0 ml 0.1 ml in a lactose broth, and it is incubated at 37 degrees centigrade for about 24 hours. After incubation, the test tubes are examined for the production of gas, that is presence of air bubble in the inverted Durham's tube. This is the presumptive phase. The next phase is confirmatory phase. All the positive presumptive cultures used to inoculate tubes of 1 ml in a brilliant green lactose bile broth. Incubation for about 48 plus minus 3 hours at 35 degrees centigrade. So the positive test tubes will again show gas production, and it is further examined for completed phase, which is the last phase. So in last phase, what you will do, you will take one loop full of suspension which is streaked onto eosin methylene blue agar. Earlier we were using liquid culture media. Now we are using semi-solid media, which is eosin methylene blue agar. In this, you take one loop full of suspension and you streak it on that media, and you incubate it for at least 24 hours at 37 degrees centigrade. So after incubation, you are going to see the greenish metallic sheen colonies which are present on that eosin methylene blue agar. You have a MPN table, that is most probable number table. You are going to count the colonies and correlate with the MPN table available. The other method which is used is single cell isolation method. In this, isolation of one cell from mixed culture and then allowed to grow separately on the other culture medium. It is also of two types. One is capillary pipette method, the other is micro manipulator method. So single cell isolation method is of two types, capillary pipette method and micro manipulator method. Capillary pipette method, the sample containing microbes is placed on a cover slip. Then each drop is searched under the microscope that contains single microbe. That drop is then sucked using sterile capillary pipette, and it is transferred to fresh medium, and then you incubate it to start the multiplication of bacteria. This is how it is done. The droplets containing microbes are put on sterile cover slip with a capillary pipette. Each droplet with a low power adjective is examined under the microscope until a droplet containing only one microbe is seen. The droplet containing only one microbe is picked up by that capillary pipette, and this droplet is then transferred into a culture tube which is containing a suitable media. Is it clear, or should I repeat? First you take a cover slip, then on that cover slip you pour the droplets containing microbes on that sterile cover slip with a capillary pipette. After you have put that, you examine that cover slip under the microscope until a droplet containing one microbe is seen. You are going to see bacillary forms which are motile, and coliforms are usually motile. So the droplet which is containing only one microbe is identified, it is picked up by capillary pipette, and that droplet which is containing that microbe is transferred into a culture tube containing suitable medium. It is a very exhaustive method. Then you have a micro manipulator method. In this you require a microscope as well as a micro pipette. What do? You have adjustment that can move in all direction. Microscope search for single microbe present, sucked by micropipette, it is transferred to culture medium, and then it starts multiplication. Again I'll repeat, you require a microscope and a micropipette. By the help of microscope, you search for that single microbe. That single microbe which is present in that droplet is sucked by the micropipette. It is transferred to a suitable culture media and then incubated for starting of multiplication. Now this slide shows the spread bacterial culture on a cover slip, and the cover slip is being examined, and then this droplet which is containing the microbe is re-sucked, है ना, using a sterile solution, and you aspirate that suspended cell into the micropipette, and through that micropipette you transfer it into a suitable culture media. This photograph is of micro manipulator method. How is it performed? This is the instrument by which we perform this test. Then you have certain advanced methods also, like micro inoculation culture method. It is a substitute for pore plate and most probable number method. It was developed by Thailand research team and it succeeded in its fieldwork. This test was found out to be much easier and more durable than the pore plate and most probable number method. So this is an advanced method which was developed by Thailand research team. Then miniaturized technique, that is micro inoculation culture plate, MIC plate is utilized. What do you do? That is a simple 96 well micro titer plate which is having a 5 mm breadth and 10 mm length and about 1.5 mm thick, and these are 96 well. Therefore 96 samples is equal to 96 petri dishes. That each well is giving you the acknowledgement of each petri dish. So there are 96 wells. That 96 well will correspond to 96 petri dishes, although it is performed in a smaller area. So 1 micro well is equal to chromo-occult coliform agar plus inoculum volume of 10 microliter is taken in that micro well. Chromo-occult is a commercially available coliform agar. You place that coliform agar into that micro well and you inoculate about 10 microliters of that sample into that each well. Then it is incubated at 37 degrees centigrade for about overnight incubation, which is 12 to 15 hours. What do you see? You see colonies and there are different colors. It is chromogenic media. It is commercially labeled, so they have patented that. So you easily see and identify through colors. If there is a dark blue color present in that well, it is identified that E. coli is present in that. If it is red in color, then other coliform bacteria may be present in that. So finally, this well bacteria is re-streaked by EMB agar medium for isolation of that particular bacteria. And then comparison is done. Comparison, how is it done? The dimension is calculated, MIC in pore plate and most probable number. So detection time in MIC is 12 to 18 hours. In pore plate it is much longer, it is 2 to 3 days, and most probable number method it is 3 to 4 days. So MIC method is slightly less time consuming. Medium use, it is used 0.5 ml per well. Then in pore plate you at least require 25 ml per plate, and in most probable number you require only 9 ml per plate. So you are seeing that less number is also being used in MIC method. The medium. Then the inoculum size. Inoculum size is 0.01 ml. It is very small size compared to pore plate method. There you are using 1 ml. And in most probable number, either you can use 10 ml, 1 ml or 0.1 ml in corresponding dilutions. So this is how comparison of these three methods is done. The advantages of this advanced method is it is rapid and accurate reading you occur. It is less costly. It is less time consuming. If you talk about cost, the pore plate method roundabout causes $36, whereas MIC costs only less than $6. That includes all preparation, media, disposable material which we are using, other utility materials, etc. And this also reduces the incubation time. As I told you, only 12 to 18 hours are required for that incubation. And high volume of microbial enumeration sample. एक second pause करो. On karo. So as I told you, this method which we are discussing, it is miniature technique, that is micro inoculation culture. This micro inoculation culture is rapid, it gives more accurate reading, it is very less costly, it reduces the time of recovering the isolation. It is less than 12 to 15 hours. Although pour plate method, if you see, it takes 2 to 3 days, whereas most probable number takes 3 to 4 days. So on the same day you are getting the report. Whereas it also requires that high volume microbial enumeration samples are taken in this. So in this culture, there are several methods which we have discussed today, but most important, you should remember that what is a pure culture? It is single species of microbe. You should always follow aseptic technique. There are number of procedures, but you have to choose from them that which procedure will be required for you. You can use either plating method or most probable number method or MIC method. Whichever you utilize, it will recover the bacteria in pure culture form. Thank you very much for your kind attention.